Good morning and good afternoon, everyone. This is Mahalea Marshall with the IQ Consortium Secretariat. I'd like to thank you for joining this morning's webinar entitled Dissolution Method Troubleshooting and Industry Perspective. This webinar is presented by the IQ Dissolution Working Group and is sponsored by the IQ Analytical Leadership. I will introduce Carrie in her stead. Carrie Gutant is a Senior Director, Analytical Chemist with 20 years of experience in drug product development spanning from first human dose to commercial launch. She has expertise in product performance of oral dose forms, including dissolution and disintegration method development, analytical control strategy, biowaivers, and applied in silico modeling of in vitro dissolution. She received her doctorate in analytical chemistry from the University of Michigan.
Thank you, Mahalea. Hopefully Carrie will have been able to rejoin us by the Q&A session, but we'll go ahead and make a start. So we have several speakers today that will be going through the various areas of dissolution method troubleshooting and 10 case studies. And you can see these people on the screen. The shared slides will include the full bios of all the speakers today. So I'm going to get us started off. So on the introduction to dissolution method troubleshooting. So I imagine the audience is very much interested in dissolution, but for those that maybe it is not their main area of interest, dissolution is a key product performance test and quality specification for almost all solid oral dosage forms. And with that, it brings an incredible amount of regulatory scrutiny from not just CMC drug product reviewers, but from biopharmaceutics reviewers, conform reviewers. So probably out of all the tests on the spec for a solid oral product, it's probably one of the most looked at. Obviously, the development of a method will focus on determining conditions that can discriminate for potential critical material attributes or critical process parameters. And since maybe the last 10 years, there's been a real driver to link that in vitro dissolution to the in vivo product performance and the advent of so-called clinically relevant dissolution methods and specifications. We look to balance the discriminating capability of a method because obviously an under-discriminating method could pose a patient risk that bad product could make it into humans. But essentially an over-discriminating method could also lead to a patient risk because if you cannot release drug to the patients, then they could result in a lack of medicines available to the patients. So it is a careful balancing act between supply risk and patient risk, and obviously there is also a risk around method robustness in there. And so with all of this and this careful balancing act, the method will move through an analytical life cycle from development into commercialization and onwards. And often the sources of variability that your method will be exposed to will increase during that time. And quite often you will see scenarios arise which will require a method investigation. Some of those scenarios that lead to investigations are your classic, where you get an out of specification, your OS. Equally, you're out of trend results, be that on a stability study or such. Maybe you will see increased variability in your results over your historical data set that you might want to investigate. Potentially, you will see increased progression to stage 2 or 3 testing that hasn't been seen historically. These could lead to investigations. Potentially, you start to see observations of issues during the buffer preparation. The buffer is, as Christian will outline in the next section, a key element of any dissolution method. Potentially, you start to see unusual visual observations during your dissolution, such as coning, floating, dancing, these kind of things. Maybe you've had an analytical tech transfer that hasn't gone well, and you've shown non-comparability during method transfers, and that can be between laboratories or just between different equipment types. And this would also lead to investigations. And then if you try to introduce automated dissolution equipment, that can also lead to issues arising in your method that could lead to be investigated. And we will probably have case studies that address several of these today. If we move on to just the investigations themselves, often you can think of a dissolution method into 3 distinct activities. You have the procedure that you would do to obtain the sample for the analysis, which is obviously the dissolution apparatus part itself, the USP1, 2, for example. And then you have the analysis of the sample, which could be by UV or HPLC most commonly, and then the calculation of the dissolution result. And in order to break this down, the team, the IQ team that's presenting today, when we address the manuscript, we often found that a very good way to look at this dissolution method is to break it down into the fishbone and look at each element, each arm of the fishbone individually, and what areas can you see issues, and we will have some case studies related to these various arms. And those arms are obviously in no particular order. You have materials, so what goes into your dissolution test, we'll cover them, the method itself, the equipment type that you're using, the measurement, the people, which can often be a very important part of dissolution, the environment that you're conducting the test in, and all of these could be contributing to a dissolution method issue. So that's the introduction. I think I'll hand over now to Christian, who's going to talk to you about the materials. So Christian, over to you.
Thanks, James. Just have to make my comment. Thanks for the introduction. So the first part of the fishbone diagram that we'll be going to describe within this webinar is now the materials part. And in general, the materials in a dissolution test clustered into groups or sources of variability as indicated on the slide. In the following, we will explain potential source of variability deriving from drug product itself, the reference standard, and the dissolution medium, obviously a big source for variability in the dissolution test. So we can go to the next slide. So this is first on the drug product and the sample and the reference standard. And before starting a comprehensive investigation on the dissolution test or on any failure, it absolutely makes sense to think first about the drug product and the sample itself. And in this regard, the first question that might come from a project or from your team and yourself will often be, is the observed issue a method related or a sample related issue? Most likely, it can be quite helpful to test a reference or a well-known sample to exactly address this question and to exclude any method-related issues. However, before one should start performing additional tests, it may be more straightforward to first ensure the correctness of the sample that was tested, for example, appropriate labeling, storage, and manufacturing, which in turn then potentially triggers further investigations on the aspects that I just mentioned. For the reference standard, one should absolutely carefully check the identity of the reference standard, the purity and potency conversion in case of salt forms or any co-crystals of the reference material. The largest part on the next slide, we can go to the next one. The largest part of the materials section is obviously the investigation of the dissolution medium itself. And here it is essential and very easy to check the correctness of the reagent weights and grades. A very prominent example is the use of a phosphate salt, which can be present in different hydrated forms, which can finally lead to deviating weighings in case the wrong hydrate is used for the calculation or for the weighing itself. So if you use a wrong hydrate form and the pH is afterwards adjusted, this can result in an overall different composition of the dissolution medium. And here it would be a quick win to have a clear expectation of the pH prior to pH adjustment. In case of anhydrous forms, that's also true for many salts used for dissolution media preparation. And in case this anhydrous form is not stored properly, water binding can occur and therefore also associated weighing issues. And this must be considered as well for the very basic steps of medium preparation. On the other hand, this will be also highlighted in the course of this webinar. When you use bulk media or concentrates, the adequate mixing and mixing time and also the dilution to the final dissolution medium must be ensured. And as said, this will be highlighted in the case studies 1 and 2 in this webinar. Further, the quality of the reagents must be considered. For example, the microbial contamination or any potential cross-contamination with other reagents that may result from previous analytics, for example, from inadequate rinsing of the equipment used for the test. In case enzymes are used, for example, when you observe cross-linking of gelatin capsules, this needs very careful consideration. In this case, USP states a quantity of pepsin that should result in an activity of not more than 750,000 units per liter. And therefore, the correct calculation of the mass to be added to the dissolution medium is of special importance. And here the COAs of the USP grade of the enzymes need to be carefully considered. At this point, we want to highlight that it's important to also have a closer look on how the pepsin unit is reported in the COAs. For example, it can be that it's reported as a percentage protein and pepsin units per milligram protein, or directly as pepsin units per milligram product. Last but not least, surfactants, they are quite often used to increase solubility or solubilization of poorly soluble drugs, but therefore they also offer a further source of variability in the dissolution test. For example, there can be incompatibility between a prominent surfactant SDS and potassium ions, or there can be also varying grades and purity that in turn finally can result also in impaired solubilization capacity, which will be highlighted in case studies 3 and 4 for SDS. And with that, we can jump into the case study number 1, and that's one fitting to the material section, as already highlighted by James. And the first one is the buffer preparation from a concentrate. In this case, we had in the case study the dissolution test of an immediate release tablet formulation. It was a basic NCE molecule, and the dissolution test was conducted in 500 milliliters of pH 5.5 acetate buffer using the USP apparatus 2 at 75 RPM, so quite a standard test condition for dissolution test. The procedure looks as follows. For the medium preparation, due to increase efficiency in the lab, there is an instruction to prepare a 10-fold concentrate of the buffer, and then to be quicker for analytics to just dilute it with water to the final buffer concentration just prior to every analysis using a media preparation system. On the right-hand side, you can see the amount dissolved versus time for a method development batch where the development was based on. That's the black solid line. And you can see a further profile at the very bottom of this graph where we see an out of specification event for the first intended batch release analytics. So investigation started, and investigation quite quickly revealed an easy case, I would say, that the final one-fold acetate buffer at pH 5.5 was again diluted 1 to 10, so there was a double dilution step in this analytical series because the operator assumed that it was still a 10-fold concentrate. So it was quite easy to start a retest because there was an appropriate justification for initiation of a retest according to a classical investigation. And here the operator used then the correct buffer, and this could finally meet the acceptance criterion of QA after 45 minutes. However, there was still a slight difference, as you can see, or already a bigger difference that led to further investigations where it turned out that there was a difference in granular particle size distribution observed. I think this example, this case study nicely describes that the use of a buffer concentrate can add for sure a source of variability. However, the time and the resource benefit of this procedure is deemed to compensate for this potential error. And in addition, in case of well-designed control measures like audit trails and thorough documentation checks, this can be quickly identified and lead to successful investigations, in this case of a retesting.
That brings us over to me. I'm Nick Zabarenko from Eli Lilly and Company, and I'll talk about a couple case studies focused on dissolution media. So the first one will focus on mixing of the media. So next slide, please. In this instance, we had a drug product with the dissolution test in pH 5.5 citrate phosphate with fairly typical profiles, full release somewhere around 60 minutes, and not much variability from batch to batch. It was a fairly robust method and production. But during primary stability, we had issues with several of our tests showing very low results relative to what was expected. Now, the media preparation in that study was done with large batches at a time, so about 150 liters of media at a time. And in every case, the media had passed spec upon its production or preparation. So we started with our fishbone diagram and started looking through all of the various elements of dissolution that could explain the low dissolution results. And if you go to the next slide, when we laid out the results in chronological order, we noticed that all of the low flyers happened in one specific preparation of media, and they were chronologically trending. So with time, each subsequent test in that media gave lower and lower results for the most part. And then when the new media was prepped, it was back to normal. So it was not correlated to anything in the drug product, was not correlated to anything in the equipment, but it was correlated with media. So testing all the various things related to media in the fishbone diagram to see what could explain such a drop in performance, we made a few development lab small-scale studies making media that would pass pH, but would not follow the recipe. So for example, making media twice the concentration of the buffer, or making media that's 1/10 the concentration, so very dilute. And twice the buffer gave us slightly faster release, whereas dropping the buffer concentration to 1/10 gave us profiles that were very similar to the ones we were seeing in the actual stability study. So that gave us an inkling to look at the media concentration. And I will say all of those preps passed pH check within the range specified. So if you go to the next slide, we were able to take aliquots of the media from the dissolution samples and run additional tests on it, including conductivity and osmolality of the media. So as you can see in the figure, osmolality and conductivity should all be within the green band as well as the pH throughout the media. But it started out high at the initial points where the media was used, even though the pH was passing spec. And as media was used subsequently, osmolality of those samples and conductivity went down, trending exactly the same trend as the dissolution performance. So that told us that A, the media was getting more dilute as time went on with additional samples from that media, and that more dilute media resulted in out of spec dissolution tests. So if you take this at face value, you can see that pH as a test does not necessarily tell you that the media is prepared correctly or if it's mixed well. Other tests may be necessary to confirm that. And osmolality can be easily calculated for buffers, and it's much more sensitive than pH, the proper concentration. It's also a fairly quick and easy test. So if you go to the next slide, we were able to do some simulations for the mixing to see if we can explain what was going on. Because the hypothesis was that as material was added to the mixing vessel and then water was added on top of it, mixing was insufficient to fully homogenize the media. So you had more concentrated media on the bottom and more dilute as you went up in the height of the tank, where the spigot is on the bottom of the tank, and the initial samples were overly concentrated in the buffer, and subsequent tests used more dilute media. So a Visimix simulation showed that in the setup that we were using, the 55-gallon drum, or rather our contractor was using, at the assumed agitation rate, first, there are some suggestions for the minimum requirements for the impeller diameter and the impeller height. So for a vessel of that size, the simulation said that you should never use a stir bar. Anything that's directly on the bottom of the vessel would almost never give you a full mixing without incredibly long mixing times. But an impeller raised about an inch off of the bottom at about 1/5 of the tank diameter, the impeller diameter being at the 1/5 of the tank diameter, would give adequate mixing after at least 2 volume turnovers, which in this system would take about 2.5 hours. So looking through the media preparation lab notebook entries, the mixing in this particular preparation was 78 minutes. So it was right at 1/2 of the needed time to provide adequate turnover, which explains quite well why we were seeing a gradient in concentration for the media during its use. So if you go to the next slide, the conclusion that we drew from that was that mixing is quite important, as one would imagine, and to achieve adequate mixing, one must consider impeller size, its position in the vessel, and mixing time. And for the 200-liter vessel that was being used for this study, the calculations indicated about a 1 minute per liter mixing time that would provide adequate mixing in that system. Additionally, it showed us that pH is not a good indicator of properly prepared media. So if media is prepared at a large scale, either the pH needs to be checked at every test that it's for every amount of media that's being taken out for a given test, or conductivity or osmolality can be additional useful measures to confirm that the media is prepared correctly. And of course, a method robustness study should be done to indicate sensitivity of your dissolution profiles to the variations in the buffer composition. So if you go to the next case study, another component of media preparation is the vessel in which it's prepared and potential contaminants. So on the next slide, this case study involves the same drug product as the previous one. And again, typical profiles were rapid and low variability, but in a different stability study, we observed above normal variability for a subsection of tests. So of course, we took our fishbone diagram, tried to see if it's linked to anything about the stability itself, time points or packaging or anything with the API. But if you go to the next slide, we were able to eliminate all of the drug product or drug substance related fishbone elements. And again, looking at the media and media preparations, we were able to ascertain that the most variable sections were unique to several individual media preparations. So particularly, yes, media 5 and media 9 gave us the worst performance. So it was again, not correlated to package, to storage, to drug or to drug product strength or to anything else. So we then started to look again into what made those particular media preps unique compared to any of the others. So if you go to the next slide, all of those media preps were made following a different, a specific cleaning procedure, which included the use of SLS. So knowing that surfactant can be a factor in dissolution performance, and this method does not use surfactant, we also knew that in this particular case, the API can bind to various surfactants due to its molecular nature. So we ran tests again in our development lab to see if media prepared with different amounts of SLS could explain such a performance. And even at 0.1 ppm of SLS, there is a marked and noticeable decline in dissolution performance with a 10% drop at the 45-minute time point that we were looking at. And by 10 ppm, you could actually see precipitate forming in the standard, and you can see release that will never go to completion. So even if you extend this dissolution test out to multiple hours, it still doesn't reach above 90%. So we were able to confirm that the hypothesis is valid, and we're able to obtain aliquots of the media used in those particular tests that gave us out of spec behavior. And LCMS with high resolution was able to measure the presence of SLS at 0.3 ppm, which puts us right in line with about the 10 to 15% decrease in expected dissolution behavior. So we knew that this most likely was caused by the cleaning procedure and small trace remnants of SLS being left behind. So to solve that going forward, we asked for a dedicated carboy here for media preparation for this project, and a procedure that included only water or non-surfactant, essentially salt-based, ion-based cleaners, to ensure that this would not happen going forward. But this indicates that when you are considering your method robustness and your dissolution robustness, if there is a sensitivity to surfactants for your dissolution behavior, it's important to consider how the media is being prepared and in what equipment it's being prepared. So I think with that, I'll turn it over to Andre for the next case study.
Thank you very much, Nick. To follow up in the general theme of paying attention to potential contamination, we have a case study about some impurities that actually came in from the surfactant that subsequently caused some oxidative degradation that I'm going to go over now. So what you can see here, one of the observations we had on our dissolution was that on certain runs and lots that we have seen is after 45 to 60 minutes, we saw a significant drop in the percent dissolved, or at least detected during our dissolution run. And this was not due to any precipitation or other effects like that. We actually did some chromatographic evaluation and saw that in these runs, we actually had quite a significant formation of a oxidative degradant of our API during the dissolution that formed. In general, we did know that there was this oxidative degradation tendency. However, it needed to be catalyzed. And in order to figure out what catalyzed that oxidative reaction, we actually found out that a Fenton reaction took place by a peroxide impurity in the tween 80 that was used in the dissolution media, and the iron 2, iron 3 that was used in the film coat of the tablet. Due to the Fenton due to the Fenton reaction, we then formed a hydroxyl radical was formed that then catalyzed the oxidative reaction in the dissolution during the dissolution test with the API and led to this low yield. If you go to the next slide, we did an investigation basically around the incoming tween to understand how can we limit potentially the peroxide impurities in the surfactant. And two of the main take-home messages you can see here in the table, different suppliers and different lots from different suppliers of tween 80 were tested. And also at initial, as well as several days after opening the bottle, down to like, in the extreme cases, to 150 days. And the general observation was that the incoming amounts of peroxide even varied between different vendors. But in general, what we have seen is that the peroxide significantly grows over time after opening the bottle. So the first step that we have taken out of that is to limit the period for using the actual surfactant to less than 30 days after opening, and therefore keeping the peroxide levels low enough in order to ensure that the reaction during the dissolution test doesn't occur. However, that wasn't enough for us from a robustness standpoint, just due to the uncertainty in lot-to-lot variabilities, that we actually went forward and, as demonstrated on the next slide, tried to actually suppress the Fenton reaction overall. And the way to do this was to actually complex the ion 3 that catalyzes the Fenton reaction with EDTA. And here you see basically in the bar graph in the bottom that with the addition of EDTA, at different peroxide levels that we actually spiked into the dissolution media, the reaction was basically completely quenched. And the two degradates that you see shown on the bottom basically did not form even after 3 days at room temperature in the dissolution sample. So basically, to summarize, surfactants can bring in impurities even at little trace levels that can react chemically with the API. And there are multiple ways, I think, in how that can be controlled through incoming material control, understanding do these impurities like the peroxide, for example, and tween 80 is known as a peroxide former, can we limit the in-use period for the incoming chemicals we're using for dissolution? And are there any other mitigation mechanisms such as the EDTA addition that actually can prevent that reaction from occur in order to get a robust dissolution method? With that, we're going over to the next part of our fishbone diagram and are going to talk about equipment, and I'll hand it over to Beverly.
Thank you, Andre. So for the next few minutes, I'll be providing a background around equipment. And equipment's a major cause of dissolution method issues. And once we have some unexpected dissolution results, there are a number of things that we want to check related to the equipment, looking to see if there's any differences from previous experiments, looking at equipment maintenance, logbooks, run reports, error logs, checking bath qualification status, any pre-run checks, such as was the temperature correct, the paddle height, etc. Also confirming the material and condition of the paddles, the sample and cannula and auto sample needles. We've had issues where we have seen degradation on metal surfaces, degradation of the API when using an acidic media, because we get metal catalyzed degradation. We've also observed if we have scratches on PTFE coated paddles, that can lead to areas where we can get degradation or sites for nucleation. And for methods that use the basket, it's important to check the mesh size and also that the baskets aren't misshapen. So these are some checks to do. And then on the next slide, one of the areas related to equipment that can cause concerns is around degassing. the presence of bubbles, so we know can impact the dissolution rate. A couple of examples are these, is we had a case where there were bubbles on the surface of an erodible tablet, and so that reduced the contact of the media with the dosage form. There can be bubbles on the mesh on the baskets, which can reduce the flow of media through the basket. And another example is air bubbles can increase buoyancy of particles. And so in all of these cases, this can affect the dissolution rate and either lead to variability or unexpected results. So if degassing is critical to method performance, you want to check the degassing equipment to ensure it's providing media of sufficient quality, and use a dissolved oxygen meter to ensure the concentration set an acceptable level. Another area on the next slide. In terms of the UV spectrophotometer, typically this isn't a source of issues if the equipment passes self-test checks, but a couple of things that we can check is to confirm that the correct settings are in place so that you have the correct UV wavelength for the method. Ensure that you're using the correct path length cells for the cuvette. And also check that the fittings to the cuvettes are secure. Any loose fittings can lead to introduction of air into the lines and failure to pull the correct sample volume through the cuvette. Another area, automation. Automation of dissolution methods and method transfers between automated systems is often a source of issues for dissolution. And this is because there's often a lack of understanding of how the automated system functions with regard to taking a sample, and this can vary between different vendor and different pieces of equipment. So for instance, we can have incorrect autosampler parameters, such as not having the correct prime volumes or purge volumes, pump flow rates, or system tubing volumes, and this can lead to issues. Incorrect settings can result in samples not being taken at the required time points because the autosampler is not able to complete all the activities before the next pull. You can also have insufficient priming and purging of the lines, so this can leave solutions in the sampling lines and subsequently diluting sample for the next time point, leading to lower results. And this is will be discussed in case number 6, which will be coming up. A couple of other points about automation. Tablets can get stuck in the sample magazine, and this is exacerbated by the tablet geometry. And so if this happens, you can end up with no dissolution profile because you had no tablet in the vessel. For fully automated systems, the valve in the bottom of the vessel may exacerbate coning for some formulations because the vessel tends to be flatter on the bottom compared to conventional vessels. And differences in equipment design from one vendor to the next or one type of equipment to the next can lead to hydrodynamic differences in the vessels that then impact the dissolution profile. And this will be illustrated in case studies 5 and 7. And so with that, I'll be covering case study 5, which highlights some differences with different automation equipment. So in the next slide. So in this case study, we had an immediate release tablet. The method was USP2 paddle, 75 RPM, 900 mils of a buffer. And predominantly had used a semi-automated system with offline UV. And then we went to use a fully automated system with online UV, and we saw differences in the dissolution profile between the two systems with the same lot of material. We looked into this, and we suspected we were having these differences due to differences in the sampling probe between the two instruments. And shown in the photograph, so on the left top photo, this is the sampling probe for the fully automated system. Below that is an image of the sampling probe from the semi-automated system, which is smaller. Yep, smaller in diameter. Since we weren't able to switch the probes between the two instruments, one of our scientists got creative and he put a plastic tube around the probe from the semi-automated system to try and get it to mimic the size of the probe from the fully automated system. And as shown on the photo on the right, we put these in the semi-automated bath. So again, where the red cursor is, that's the modified probe that mimics the size of the fully automated, and the other probe is the unmodified. So we had these both in the semi-automated bath, 3 of each, dropped a tablet. And then in this slide, you can see the various profiles, and I'll talk through each one of them. So to start, the profile at the very top in red with the X's. This is the original profile from the fully automated system for lot A, which looks very nice. Then the very bottom curve in blue with the X's, that's the same lot in the semi-automated system. And you can see there's a big difference in the percent dissolved. And also in the semi-automated system, we see coning. We see a coning effect. We get less than 95% dissolved by 60 minutes. We do an infinity spin, it goes up to 100. So then we looked at the modified probe and the unmodified probe again. So if you look at the blue line in the diamonds, so that's the semi-automated probe. This time we had to use lot B because we didn't have any more tablets of lot A. So it looks very similar to what we originally saw, which is what we expect. Then the red line with the squares, that is the modified probe in the semi-automated system. So by modifying the probe to make it wider or fatter, to be more similar to the fully automated probes, you can see that there is a difference. Profiles are faster. And so with our formulation is susceptible to coning, and we feel that the differences in the probes led to differences in hydrodynamic, which in one system caused coning, whereas in the other, it didn't. So for this particular product, we ended up changing the method and going to peak vessels to address the potential for coning. And with peak vessels, we do get similar profiles with both of these instruments. So that's one example of equipment having an issue. And at this point, I'll turn it over to Dalwin, who'll talk us through the next case study.
Thank you, Beverly. This is Dalwin Ko from Genentech, and I'll be presenting case study number 6, auto sampler settings. Next slide, please. So as Beverly mentioned in the introduction, auto sampler settings can have a significant impact on dilution results. So in this case study, we had immediate release tablets. These were from a 12-month stability time point, 25, 60, and 30-75 conditions. As you can see, the graph on the left-hand side, the 30-75 sample had a much slower profile than the 25-60 sample. At this point, we've had a lot of experience with this formulation. It's actually a B6 class 1 drug. That's why typically the dilution is very fast. We have never seen this slow down before. So the 30-75 sample was out of trend against itself from previous time pools, and also out of trend against the 25-60 sample at the same time point. So that triggered a lab investigation. What we found was that we used different equipment. So for 25-60 sample, we used a Distech Symphony 7100, and for the 30-75 sample, we used a older model, which is 6100. Another interesting finding was the auto sampler settings were different for both samples. So the 25-60 sample used a higher offset volume at 3.5 mil and a higher flow rate at 15 mil per minute, while the 30-75 sample had a lower offset volume at 2.0 and a lower flow rate as well at 10 mil per minute. Although the auto sampler model was exactly the same for both situations, there were evolution 4300 again from Distech, but the settings were actually different. So the offset volume, also known as the purge volume, is basically the volume that you use to displays what is already in the auto sampler tubing before a new sample is pulled. So too small a offset volume could leave some of the previous samples in the tubing that is basically carry over from the previous one, and resulting in carryover and low recovery issues. So that seems like a reasonable explanation. Then we went on to do this experiment. Next slide, please. So what we did is we kept everything else the same. The only change we made was to change the offset volume and the flow rate from 2.0 to 3.5 for the volume, and the flow rate from 10 to 15, basically 30/75 sample. And the orange curve you see here, that's the before, and the red curve you see here, that's the after. So you can see there's a drastic change. Basically, the dilution just seemingly become faster with this change in auto sampler settings. And as a comparison, the 25/60 sample obtained using the higher offset volume and the higher flow rate was basically overlapping the blue line with the 30/75 with the same auto sampler setting. So that was a very convincing experiment to show that the auto sampler settings were causing the difference. But the second question is, did the flow rate actually made a difference here as well? So we did another experiment using the same offset volumes, but at this time just trying two different flow rates at 10 and 15. And the conclusion is that at least within this range, 10 and 15, they made a very little difference. So at this time, it was very clear this smaller offset volume 2.0, that was the root cause, and it was insufficient. But then the next question is, how do we know definitively the higher offset volume was giving us 100% recovery? So we did another experiment. In this case, we prepared a pre-dissolved solution sample, so we know exactly how much was in that. And then we use this method. The first time point, 5 minutes, we did a pull from pure water. And then the next time point, 15 minutes, we pulled this solution sample. And we were able to achieve 100% recovery with this higher offset volume than 3.5. And in comparison, when we used the 2.0, we got much lower recovery than 100%. So this experiment was definitive to show that, at least in this case, specific case for this model, the 3.5 was sufficient to achieve 100% recovery. I do want to point out that is depending on the specific model of auto sampler you use, the offset volume may be different. So you need to check with the manufacturer. So in conclusion, it is very important to understand how the auto sampler functions, so you can use the right or sufficient offset volume to displace the previous sample in the tubing, and to ensure the sample is representative of the actual sampling point. With that, I'll turn over to the next case study.
Hi, this is Johanna Milsmann from Boehringer Ingelheim, and we also have a case study about automation. So on the next slide, please. We did have a formulation which we established on a fully automated dissolution system during early development. And then during the course of development, we had a formulation change, and we had to re-evaluate the dissolution profiles between manual and the automated systems. And as you can see on the lower left-hand side, when we drew samples manually, those were the they had the lowest velocity of the initial dissolution speed. Then we also we made some dissolution profiles with a semi-automated system and with a fully automated system, and both of them were way faster compared to the manual sampling. As both for the manual samplings, we drew samples via cannula, and for the automated systems, we had there's some tubing attached, and then there's an online measurement with a photometer. So as a first elimination of the cause of this error, we did 2 more dissolution runs. One on the semi-automated system and one on the fully automated system. And during this run, we also drew manual samplings. And as you can see, for both systems, the manual sampling as well as the semi or fully automated analysis resulted in the same profile. So tubing was not the cause of our error. So when you switch to the next slide, you can see our 3 different systems. So for the manual system, we did have a Sotex 87 Smart, which is basically just a plain glass dissolution vessel with a stain paddle, and we drew the samples via cannula, which is then retracted after sampling. For the semi and fully automated system, the Sotax AT7 Smart with the online photometer, or as well as the Sotax AT70 Smart, are equipped with a hollow shaft, and that is basically this mesh insert into the paddle itself. And in addition, we also have the bottom valve in the AT70 Smart, which is used for media replacement and cleaning. And in this specific example, our tablet was really sensitive to those additional surface roughnesses, and that increased the solution the solution velocity immensely. And for our case, that actually went back so that we couldn't actually implement a automated sampling for that product. And with that, I would like to go over to the next point in our fishbone diagram, and that is the methods. The methods are, on the one hand, basically what you write down, how you pursue a method. So it's the medium, it's the paddle speed, or if it's a basket, it's the vessel temperature. Those can be obvious causes for errors. So for example, if you would run a dissolution run with 50 RPM in a paddle setting instead of 75 RPM, maybe that would result in a faster dissolution velocity. Or also if the vessel temperature would differ from the specified 37 degrees plus minus 0.5 degrees Celsius, that is also, I guess, sort of an obvious cause for error and can easily be checked back to your method, which you wrote down. But then there are also method specifics which might not be written down, or also are more or less handed over or part of a protocol. And that is, for example, that when dropping a tablet into the dissolution vessel of a paddle run, the paddle should be stopped prior putting the tablet in, because otherwise it could be hit by the paddle and then maybe destroyed, or at least checked, and that could lead to increase dissolution rates. Also, when you do manual sampling, all of the dosage forms should not be dropped in at once, because the manual sampling is done vessel after each other, and then if all samples would have been dropped in at once, the specified time points will probably not be met for every time point you have to draw. And then for the method part, we would also like to highlight two specific parameters. On the next slide, please. One is filters. So every filter which is used should be fully validated with a couple of different points to ensure that they're used correctly and that they have the correct performance. And those parts should include that the discard volume of the filter is correct. And a common cause for error is that the discard volume is checked with the middle or the highest dosage strength, but it has to also be checked with the lowest concentration to ensure that the filter material is saturated and that you do not lose any API due to incorrect filtration. Also, the filter efficiency is really important. So if you have a too large pore size, smaller particles could migrate through, and that is easily checked by just sampling at a time point where you have undissolved material with the discard volume you have previously established, and then you analyze one sample right away and just let the other one sit for a little bit or dilute it with an organic solvent, and that then that could result in an increased content measured if you have a filter which is not efficient enough. And last but not least, some filters also have leachables in them, and you could check for that just by filtering a blank solution of your dissolution medium through that. And especially with HPLC methods, if you encounter peaks at time points which could impact also your API peak, that is important to know. The second parameter which we would like to highlight are sinkers. So sinkers sometimes have to be established for a method, and there is a huge variety of different sinkers available on the market. So for example, if you establish a Japanese pharmacopoeia sinker, and then you would use another closed sinker with a different number of coils, that could result in different hydration of your sample and hence an altered dissolution rate. So if you established a sinker at one point in your method and you have to switch it, it is really important that you do a full risk assessment and check for its impact on your dissolution method. And with that, I would go over to the next case study, which is about filter casings. In that case study, we did have a method in place for a product with a pedal apparatus at 75 RPM and a sodium acetate buffer at pH 4.5. In early stages, we did have lower dosage strengths and a lower SLS concentrations, but then we had to switch to a higher SLS concentration as we had an increase in dosage strength. We sampled our formulations with an automated sampling device, which is basically a syringe, which then pushes into through a filter and a cannula into the dissolution bath and draws its samples. And when we use the PAL Acrodisc filter with 1 micrometer filter size, this with the increased SLS concentration in the medium, this led to a lot of foaming and unequal sampling and sometimes no sampling at all. For this case, we found an easy solution because the PAL Acrodisc filter is also available just with a different casing. It's a little bit smaller and rounder, and just due to the change in the filter casing, we got reproducible sampling. Which is, in our case, really specific to the nature of this automated sampling device, because it pushes, like during sampling, it pushes air through the filter, then prevents the filter in the cannula and draws the sample. However, it's also a case study to make you aware that it's not only the filter and the filter material, but also the filter casing can have an impact on your method. And with that, I would like to give over to the next case study.
Thank you very much, Johanna, and hello everyone from my side. This is Alexandros Kourentas presenting on behalf of the Dissolution Group at Novartis. So the next case illustrated in the next slide, please. So the next case is about is the early spikes which are observed in dissolution profiles when measuring higher percentages of the drug dissolved at early time points than at the subsequent time points. And the root cause for this observation is that the undissolved material which is sampled and collected on the surface of the filter can be dissolved during the filtration process. Apparently, the significance of this effect depends on the mechanical pressure which is applied during the sampling, the filtration volume, as well as on the tendency of the undissolved material on the surface of the filter. And in this context, the IQ group is proposing 3 different solutions in the next slide. Depending on the purposes of the investigations and the suitability of the method or optimization steps, which will finally allow for validation, here the analyst may select the most appropriate solution. This could be either the use of a pre-filter which is attached to the top of the sampling probe, or the collection of samples with lower volumes. In the first case, it is strongly recommended to confirm the adequacy of the filtration when using the pre-filter by testing the drug absorption onto the filter, while application of lower volumes may require manual sampling as well as a change from the UV spectrophotometry to HPLC analysis. In case the sampling procedure is not reproducible, it can be skipped by utilizing UV fiber optics, which will allow for in situ dissolution testing. And with that, I would like to hand over to Christian, who will walk us through the next case.
Thanks, Alexandros. So the last case study will be about post-sampling precipitation. This case study is describing a case or a set of a comparative dissolution testing of an immediate release tablet formulation that was conducted at a CMO lab. The drug of interest in this case was a weakly basic molecule with a pH-dependent solubility. And according to this chemical nature of the compound, we observed fast, robust, and complete dissolution in SGF, as we already expected from the solubility data of the compound. However, as you can also see on the right-hand side, we observed also quite high variability for the two other conditions that are often tested in the comparative dissolution testing set. And here it was quite unexpectedly high dissolution results next to the high variability, because we were quite astonished about the high dissolution, considering the low solubility of the compound, especially at elevated pH values. So we started an investigation again and thought about potential sources of this variability and this unexpected results. And on the next slide, I would like to guide you a bit through our thoughts that we had at this time in the company. So we checked the procedure that was followed at the CMO lab, and we observed that despite this low and really dramatic pH-dependent solubility, there was no dilution step included. So the team missed simply to add a dilution step for this comparative dissolution study. So we first then decided to have a repetition of exactly those CMO experiments within an internal company laboratory, again without the dilution, to just confirm the dilution should be in the focus of the investigation. And as stated here, we were able to confirm the high variability and also the unexpected high dissolution values for the elevated pH conditions at 4.5 and 6.8. So our hypothesis was that the drug experienced supersaturation followed by precipitation before the analysis via HPLC. And our further thoughts were that then in turn the injection of precipitated particles that were in the next step diluted with the mobile phase during the HPLC runs caused finally this unexpectedly high concentration values and the high variability during the analytics. On the next slide, we added a dilution step, and you can see now the direct comparison between the left-hand side and right-hand side, where we have the same data set, first glance without the dilution as already presented, and then the optimized procedure including a dilution step. So we did not repeat the analytics for SGF because there was obviously no problem and there was no drug precipitation observed. And as you can see, the introduction of a dilution step, in this case 1:1 with 0.1 HCl after the filtration and prior to HPLC analytics, was already the solution and yielded then much more reliable, less variable data, and was more in the direction what we expected from the Fiscam properties in terms of solubility. I think that's a pretty nice example, and I think that's often happening. And the recommendation from the IQ group would here be to thoroughly think about the solubility of the compound, especially in media that are not the QC medium and where you may not have the highest experience for your product, and in this case to include a dilution step before the analytical finish. And with that, I would like to hand over to Fredrik for the last topic, environment, people and measurements.
Yes, so I will take on the last legs with these topics. I will start up with environment and people, and people in especially when it comes to training and use of the equipment. So we have one example in the paper where sample introduction has been done erroneously. So there was a manual approach with staggered drops with 1 minute intervals. But the analyst didn't start the rotation the paddles at the same time as dropping the formulation. So that wasn't started until the last vessel number 6. So in vessel number 1, it sort of had a soaking time of 5 minutes before the rotation started. So every subsequent pull would then have less rotation speed than expected and gave much variable results. Another thing is the loop of having the time points concluded with pull and filtration, where filtration is sort of the stop of the ongoing dissolution to see the current concentration at that time point. And that could be another case that we've seen has been where they haven't had this kind of staggered approach. So they have more or less the same drop, and then they have sampling very fast, and then sampling all 6 at this, and then waiting until the last sampling before they start the filtration of the first set. Both these are example of manual introduction and sampling. We have one example as well when it comes to a semi-automated equipment where we have an autosampler collecting in tubes. So we have cannula probes with quite high porosity, so it wasn't really stopping the filtration. So the method described that you should do a second filtration with a syringe filter, but the approach was to wait until a non-designated time to do the second filtration from the autosampler, which then gave quite high variability in the results. Something I'd like to stress as well is all this visual observation that you can do with a dissolution, where your tablets, capsules may behave not as expected and might explain your out of specification or out of trend results. And that could be if you have not sufficient degassed media, you can see bubbles forming on your formulation. That might even give you floating formulations, or that you have formulation that is sticking to the vessel walls or to the rotation device itself. Apart from sampling, you also have the environment where your equipment is located. And if you have other type of equipments that generate vibration, we know that vibration is one of these kind of things that we like to avoid when it comes to dissolution. So if you have other equipments that generate vibrations, they should not be in conjunction to your equipment. I know of one case where they had these on and off higher results, and they had when they did the troubleshooting, they finally found that in the next door room they had a vacuum pump that was connected to an MS system, so it was intermittently going on, and if there was a dissolution going on the next side of the wall, they sort of could be affected. I also had at my site here, we actually had construction work just outside the building, and we had to negotiate with those construction workers to have a period of time where we could perform sensitive dissolution analysis and don't have affected by vibration. So we can go to next one. Then we have for measurements of your data. If you have a profile that is consequently lower or higher than expected, something that could go downward could be your measurements that something is off. And then you have to go down and check against your method parameters so that you have the suitable weighings, suitable volumes, right type of media, that the standards have been prepared accordingly, that you haven't mixed up with different volumetric flasks, for instance, when you're preparing those. And also that you have used the correct instrument parameters and double check that. And that also has been used in the calculation. So if you have used that and that matches up. And then also for the calculation, that you are using validated tools for calculation, either that you can buy off the shelf, or if you have developed them yourself, that they are validated appropriately and documented, so you can show that to authorities if you have any, if there comes any questions. Data we produce, at least early development, we might use quite a lot of type of corrections where we have weight corrections or normalization because we don't know the actual strength of a granulate produced, for instance. And then it's essential that the profiles and data you produce, that you label them accordingly, that they are normalized, so anyone who uses the data can take that into account. Another issue, something that you might take into consideration, is how your sample and standard is stored. If not online analysis, but offline, and you do that the next day, and you know you have the stability of samples and standards, and make sure that they have been stored accordingly. So if you have room to temperature stability, that's fine. But if they have then been placed in a refrigerator, then you might have precipitation of your sample. And it could be the other way around, that you need refrigerated of your samples, because in room temperature you might have some degradation instead. So there are some few highlights. I go over to James.
Thank you, Frederick. So we're on the home street now, so I'm just going to give a few concluding remarks. So I'd like to thank the IQ Brain for showing all the case studies. Let's just go to the next slide. So I think what is clear from the case studies, and if you saw some of the fishbone diagrams on Nick's examples, dissolution methods are absolutely multivariate. And I think without establishing appropriate controls during your method development, you could quickly get yourselves into very difficult situations, because without those controls, you may find that your results are not reflective of the product performance that you're actually trying to study. So building that firm understanding of the method, the equipment, the materials, the measurement, the people and environment is required. So having a firm handle on your laboratory and your product and the method is key here. If you allow to minimize that variability when you're developing your method in the sense, and allow for that enhanced product understanding, it may avoid costly investigations later in the product life cycle, where inevitably your method will be exposed to more variability, more analysts, more laboratories, more batches of dissolution media, more batches of input materials, more batches of actual drug products. So if you understand it, then you can quickly know where the likelihood is when you see a method failure, and that could be sensitivity to gassing, sensitivity to sinker design, whatever. But if you know those, then you can quickly direct your method investigations to those key failure points and have established proper controls. So just a few best practices to sum up. I think there's nothing to be said. There's a lot to be said for investing in your people. I think having a well-established dissolution training program is key. I think some labs train by, the last person in is trained and then they train the next person in. That Chinese whispering approach of dissolution method training is not a great one. You should have experts training to a set program. You should assume that people who come in have no background, because they may have bad habits, they may have, if they have done it at other laboratories, you can never really trust that they will do it as per your own company's SOP. So having that training program is an absolute must. You have to understand the capabilities of your equipment portfolio. So I think Davin's example is a great case of that, where there were subtle differences in auto sampler settings and they led to big differences in dissolution profiles. So understanding what your equipment is capable of, also translating the terminology from the different manufacturers. If one calls it prime volume and the other one calls it offset volume, these are different terms, but they may mean the same thing. There's also a term if you use Agilent technologies called prime loss volume, and it's very key to understand that and make sure that is set correctly and in your configuration file rather than in your method. I think Christian mentioned introducing appropriate quality control checks, as is audit trails, documentation checks to make sure that what you're doing is correct. Hopefully all labs would have that if they're operating to some level of CGMP. And I guess the other part, which is clear and it builds your method understanding, is if you can prioritize during development well-designed robustness and ruggedness studies to actually understand what your methods are sensitive to and then introduce the appropriate controls for them. So I think with that, we are now finished and we will hand over to the questions, but I think it's just worth flagging thanking all the speakers, but also acknowledging these other individuals on this slide who contributed to the manuscript, which has been published in the November issue of Dissolution Technologies, and they are all contributed to the case studies as well. Might be worth also pointing out that the case study numbers in this webinar do not match to the case study numbers in the manuscript. We rejig the order slightly in order to allow a better flow of the webinar. And with that, I will hand back over to Carrie for the Q&A.
All right, thank you. And I'll remind everybody that's listening that you can type your questions into the question box that's part of the WebEx setup. So the first question I have is about whether we are going to share a copy of the slides with the listeners. And the answer to that is yes. We are planning, Mahalea will be sending out both the slides and also a link for the recording of the webinar. So everyone should be able to access that and also be able to share that with interested colleagues who weren't able to make the call today. The next question I had was about the work that you presented, Nick, in the case study where the pH of the media was found to be part of the problem. Could you describe a little bit more about how conductivity and osmolality were measured?
Certainly. So conductivity was actually measured with the pH probe. The pH probe that was used had the ability to measure conductivity. It was just not part of the normal SOP to record those measurements. But the same probe was then used for those aliquots and was able to provide those values. Osmolality, I forget what the name of the device is, but essentially it's a device where you take a small sample on the order of 0.5 milliliter into a cuvette, and it measures osmolality through osmotic pressure measurements with the internal setup. And I can look up what it's called, but it's a fairly simple test. It's a few seconds per sample, but it's a device that I think is dedicated for that particular measurement.
Yeah, I think it's called osmometer, and I think if they're obviously not that common in dissolution labs, but I think if you have any colleagues who work on parenteral products, they will have access to one.
Yep, that's exactly what I was going to say.
we did have to go to our bio R&D colleagues for that measurement.
All right, and the next question is for the SOTAC's automated dissolution case studies. The question is whether the rapid dissolution profile has been observed for other drug products when comparing the fully automated and semi-automated dissolution testing.
No, actually not. So basically, we had other products where we had coning as described in the other case study, but I guess that was a special case. And that's also why we blamed the tubing first.
Yeah, and I'll just add, in our own lab, we have had a case study for one of our products that wasn't shared as part of the paper, but we did observe a similar phenomenon where the additional resistance caused by the valve covering at the bottom of the vessel did cause an increase in coning and a slowdown in the dissolution profile for one of our products. So I think we have seen that in multiple cases, and in response, I think the vendor has made some adjustments to the design of their next generation system and also offers some other options for designs of the valves on the legacy system.
Yeah, the next generation SOTAC's fully automated systems don't have the valve in the bottom of the vessel. They're more like the G3 multi-dose edition coming from the top.
Yeah, and they also do not have the hollow shaft anymore. So basically, the new versions all eliminate those problems.
You can choose to have them with the hollow shaft, but the most common specification is to have the cannula. Yeah, and I guess that is, if you're thinking about transferability from different systems, then having cannulas that are on an autolift type system are actually far more transferable across system types.
Yeah, and I guess in that case, if the cannula itself is causing a change in the hydrodynamics, then that'll be more consistent from device to device. All right. I noticed in a lot of the case studies that we shared, the symptom of a problem looked very, very similar. In almost all the cases, except for the one at the end, the dissolution profile just showed some suppression. I wonder if a few of the speakers would be willing to talk through a little bit more of their thought process as to how they did narrow down the possible causes of the slower dissolution that they observed.
So I guess I can start us off, and not to belabor the point, but the fishbone diagram is very useful in these discussions because we just start looking at what are the possible ways that dissolution could slow down, and then it's a matter of isolating the profiles or the material that went into the tests that generate those profiles to figure out what's unique about them relative to everything else that generates reasonable profiles. So we typically start with the drug product itself. Has something changed? Is there something particular about that batch of material or that particular test, the way it was performed? And if we don't isolate it to the drug product, we then start looking at the dissolution test itself, and mostly start with whatever documentation exists and go through it to see exactly where we can pinpoint the difference. If we can group it in a way that would isolate it to, for example, media prep, that becomes a little bit easier because then we just focus on the media. If we can't, we often will isolate it to a specific time frame, and then it's a matter of figuring out what happened in that time frame that was unique. Different operator, for example, or different temperature that day in the lab. In these particular cases that I've shown, it was media in both cases. So because we isolated to media prep, then just became a matter of seeing, well, what could shift the profile that much. SLS, for example, could not shift the profile down to the levels we saw in the first case study. You'd get precipitation and it would just plateau rapidly. It wouldn't be a slowdown like that. And you would see precipitation at those levels. They'll be fairly obvious. An analyst who was running this for days and weeks would not miss that. Whereas making media with different concentrations showed that that certainly is a strong possibility. So that gave us a hypothesis to pursue and to be able to test. So.
Thanks, Nick.
Right,
I think the point at looking at the drug product first is always an interesting one because we're analysts and we will look at, we'll say we've done our method right, and the formulators will say they've made their tablets right. But I think it's important to remember that dissolution methods can often be discriminating, and that can often be the what we're looking for. And so it is. You know, we focus very much today on dissolution methods and things that can go wrong with dissolution methods and how to investigate them and find the root causes. Equally, things can go wrong during tablet manufacture, and therefore sometimes you may do your level 0 lab investigation, and if that doesn't find anything immediately obvious and immediately identifiable lab error, you may then start to have some conversations with your formulation colleagues, and then only then once it has got to that point where they are saying that there is nothing immediately identifiable in the process, you then maybe move on to that really in-depth look across everything to see what has actually changed.
All right, thank you both for sharing your thought process there. I have a couple more questions that came in while you were discussing that. One was around the case where the surfactant introduced impurities that led to oxidation of the drug substance. The question is whether there's other surfactants that could also potentially lead to oxidation, or maybe even other sources of impurities that could cause degradation in the sample.
Yeah, maybe I can chime in there. I think it's important to understand in general what potential impurities you can bring in with your surfactants. What kind of grades of surfactants can be used? We have seen quite a big difference, right, in some lower reagent grade material. But in general, the polysorbates are known to have that peroxide impurity that can grow on stability. But I would, in general, take a good look at grade of surfactant that's being used. We have seen other impurities as well with SLS. I haven't seen a lot of those issues in the past, but if we're going a little bit more uncommon surfactants, in Triton, I have seen some, at least chromatographically, a lot of impurities that come with it. They didn't cause degradation, but that's definitely something to keep in mind. And I don't know if anyone else on the panel has some experience around that.
I've certainly seen tween issues, actually for a long-acting injectable, where the tween wasn't actually stable during the length of the test, and it was being added to PBS, and PBS has not got great buffer capacity, and so as it was degrading, it was impacting the, because it was in there at a very high concentration, it was actually impacting the pH of the media, and so there was a pH drift, which was then impacting the solubility. So it was actually quite a kind of a cascade effect across a 10-day experiment. So yeah, definitely the key there was that wasn't really a suitable suitable surfactant unless you added something to mop up the peroxides. And in our case, we added an antioxidant to the dissolution media, BHA, I think we added to it to prevent it degrading during the course of the test, which was a successful mitigation. But yeah, that's a real problem. The other one I've seen happen is incompatibilities with surfactants. You mentioned the SLS and potassium ions, but I've also seen people using CTAB with HPMC-AS, and they can form a charge complex which can impact the dissolution as well, just because of where their pKa's are and their cationic excipient versus cationic surfactant versus the HPMC-AS polymer, which takes on a negative charge above about pH 5-6, something like that.
I think it's worth mentioning also that the materials used when you're doing dissolution might impact your dissolution. So some APIs are sensitive to metals, for instance, for metal-induced catalysis degradations, and also the materials of the sampling that you used. That we had one old example that I recollect now, where we used some glass syringes with a brass fitting. And the short period of time where you would draw on the sample went through that brass fitting, it sort of it induced degradation in the sample, so we could see that we had very short stability of the samples. And the water source might also be something that might, if it's not purified in the correct way, might have sort of metal ions coming from there and sort of also generating degradations.
Very good. I think that brings us to the end of our time for the webinar. So I'd like to thank everybody for attending and thank the presenters once again for an excellent presentation. And again, remind you that we do have a paper that goes into some more detail and a couple of additional case studies that's recently been published in Dissolution Technologies Online. Thank you.
Thank you all for joining. Have a great day.